Hi, everybody. I'm Lizzie. I'm a third-year PhD student in genome sciences, and I am co-mentored by two PIs, one who is here, Evan Eichler, and another who is at Fred Hutch, Jesse Bloom. And today, I'm going to tell you about my favorite gene and talk about how this gene is important in your respiratory health. All right, let's see if this moves forward. Awesome. Okay, so let's start with a hypothetical situation. So say you and your roommate are both genome sciences PhD students, and you work very hard all week. And you're very excited because on Friday, one of the older students is hosting a house party. So you're going to go have some fun and let off some steam. However, the two of you show up as well as someone with an active case of influenza. And we all know very well that influenza is very contagious. So everyone gets sick. All right. And you spend the next week feeling absolutely miserable and horrible. But your roommate, who does get sick, seems to be pretty much good to go after a day or two and is back in lab by Monday. And so you're laying in bed feeling horrible and thinking, why is it that different people can have such different disease outcomes? And of course, this is probably a question you've asked given the last 5 years of what we've experienced across the world, right, with SARS-CoV-2. So as I'm sure you can imagine, this is a hard question to address for any number of pathogens. And that's because there's just a lot of factors that play a role in how infection operates in your body. So on the viral side of things like the specific variant that infects you of a given virus or pathogen, as well as just the sheer number of virions that can infect you at a given time could play a role. But then on your side of things, as a host, the relative state of your immune system at the time of infection, like whether or not you're already sick with something, maybe you have an underlying respiratory disease that puts you at risk for severe infection. And then, of course, like Stephen suggested, all of the many different genes that harbor many different variants in your genome that could predispose you to severe disease. And so as a human geneticist, that last part is the part that I'm the most excited about. But when we try to narrow in on that, it becomes also very difficult because, again, there's 21,000 genes, and the current estimates for the genes that are part of your immune system are upwards of 1,500. So how do we narrow in? Well, I think that we should narrow in on the innate immune system because the innate immune system really provides that first line of defense against all kinds of pathogens. And in particular, I want to narrow in on the airway epithelium, which is the tissue that lines the lumen of your lungs and is really the first line of defense. Those are the cells that come in direct contact with a virus when you inhale it. And in particular, there's a special type of cells called goblet cells that produce mucus. And we all know what mucus is. We've all, I'm sure, been sick and have produced a lot of phlegm. We can intuit that mucus is very important, right? But really its function is to trap pathogens and then help you to clear them through coughing. And so these cells produce this protein called MUC5AC that is really the primary protein constituent of mucus. So MUC5AC is my favorite gene for a number of reasons. First of all, it has a very unique genetic sequence. So it has this massive central exon composed of what's called variable number tandem repeats, or VNTRs. And VNTRs are simply chunks of repetitive sequence that have no unique sequence in between them, all right? And what's more is this VNTR is coding. That's really uncommon in our genomes. And it encodes for many serines and many threonines that at the protein level are decorated with all kinds of sugars. And these sugars allow for the mucin to bind directly or at least inhibit pathogens from moving through the mucus and infecting the underlying cell layer. Right? So this VNTR sequence is really critical for protein function, but it's also repetitive. And if we know anything about repetitive sequence, it's that it's highly mutable, right? So single nucleotide variants have a much smaller mutation rate relative to repetitive sequence. So that means that my two versions of MUC5AC probably look a lot different from your two versions of MUC5AC, right? There's a ton of variation that exists out in the world. So then you think, okay, well, let's just sequence a bunch of people and see how much variation is out there. Well, it's not the easiest task. And this is because still the predominant way we sequence genes is through short read sequencing. Now, short reads are great for genes that aren't structurally variable, but when it comes to something like MUC5AC, the reads are just too short to unambiguously assign to a given section of that VNTR. Right? But if we use newer technology like long reads, the reads are long enough to span most, if not all, of that locus. And so we can unambiguously assign sequence and finally resolve genetic variation here. So my hypothesis is kind of a simple one. It's that there's genetic variation in MUC5AC that exists. This variation is probably extensive and probably meaningfully variable because this gene encodes a protein that is absolutely critical to pathogen entrapment in your lungs and is important for the innate immune system. So I've done a lot to look at this gene, but I'm just going to tell you three little things today. First, we're going to ask the question, how much variation exists, specifically in humans? What might this variation mean within the context of a protein? And then what does this sequence look like in non-human primates, specifically non-human apes that are our closest living relatives? It's kind of interesting to look at. So the sample set that I'm working with is a relatively unique one. It is from these two big sequencing consortia, and it includes over 100 people that have been sequenced with long reads, specifically with this technology called PacBio HiFi. You don't need to know the details of this. It's pretty complicated, but the shtick with PacBio HiFi is it's really accurate. It's basically as accurate as short reads. It's just super expensive. And we sequenced with high coverage, which means that we resequence the same gene over and over and over again so that we can make sure that we don't have errors in the sequence that we're looking at. And then also you'll notice from this pie chart that there's an overrepresentation of African genomes. The reason here is twofold. One, we know from just surveying genetic diversity that African genomes are more diverse. And so if we sequence more Africans, we're going to find more diverse versions of MUC5AC. But also, we've grossly underrepresented African genomes in genetics for such a long time. And so these kinds of consortia are trying to fix that problem, right, by sequencing more diverse people. And then, as part of the Eichler lab, I also have access to many non-human primate genomes, sequenced with the same platform, same quality. So I can see, is there anything that's very specific to us relative to like chimpanzees or bonobos? Okay, so question one, how much variation exists? So oftentimes geneticists will first start by constructing a phylogenetic tree. So some of you may have heard of this before, but a phylogeny is simply a graphical representation of the evolutionary relationship between alleles. So I'm going to take all of those haplotypes from those human genomes, and remember, we have 2 copies of each gene, which means 2 haplotypes, and I'm going to construct a tree to figure out if there are groups of MUC5AC variants that are more similar to each other. And those groups we're going to call haplogroups, all right? So here's the very complicated tree. But the shtick here is that I can group human alleles into one of 3 major groups. You can kind of see them with those colors. So I'm going to call them H1, H2, and H3. All right. So 3 groups is kind of what we're boiling all of this down to. So what does this mean, 3 groups of genetic variants? Well, I can, because I have these long reads and I've assembled the entire locus, I can take the exon and intron boundaries and predict the reading frame computationally, right? So this is not sequencing protein, this is just seeing the sequence and calling the reading frame. And from here, I find 16 distinct protein variants of MUC5AC. And you know, if you've looked at just like the general size of proteins in the human proteome, these are massive. These are some of the biggest proteins in your proteome. And what's more is that the third most common variant down here is almost 1,000 amino acids longer than the 2 other most common variants. So these are big proteins with a lot of variation. And what I'm showing you here is actually the protein domains in that large central exon. So there's a ton of variation in there that's not even just due to the VNTR domains. So those VNTR domains I'm showing in gray. And what we think is that perhaps if you have more VNTR domains, then you have more glycosylation, right, which is all those sugars, that then could lead to an increase in the interaction with both pathogens and water. So that could make the mucus more sticky. But then also if you have excess of cis domains, which are these like ovals that I show here, if you have more of those, then perhaps that allows for the protein to fold more compactly. And so that might lead to a more rigid mucus that is difficult to clear. Right? So these are not functional assays, but these are just inferences that we can make based on just the genetic architecture alone. Okay, so what does MUC5AC look like in non-human primates? It's kind of crazy. So long story short, if we look at just the total length of that VNTR containing exon relative to the amount of VNTR sequence space we have, humans are abnormal. We have really short versions of this gene relative to basically everybody else. And the further you go back in the phylogenetic tree to the common ancestor, the longer the allele is. So you'll see the orangutan versions are absolutely massive. I'll tell you, they encode proteins of almost 8,000 amino acids. That's insane. All right. So something is probably happening here to keep our variants shorter. Maybe there is a natural selection pressure to keep this gene shorter in us. And I talk about that in my paper. So if you want to learn more, here is the preprint version, and it'll probably be out relatively soon, but we can also talk during any kind of Q&A session. So to go back to my original question, while you're laying in bed feeling sick with influenza, why is your roommate already recovered and you are not? Well, perhaps you have a different diploid combination of MUC5AC than they do, and we couldn't figure this out until long-read sequencing had been developed. All right. So with that, I'll take any questions. We have time for questions? Like 2 questions? Okay.
Hi there. This may not be super in your field, but I noticed with the gibbons, I believe it was.
Yep.
They're
a little different.
Yeah, they're a little bit different. I was just curious if you guys had any thoughts about that, considering like the orangutans seem to be much higher in length.
Totally. So for people who are not super familiar with kind of how primates are related to each other, so the gibbons are apes, but they're not great apes. So us, chimpanzee, bonobo, gorilla, and orangutan are the great apes. And so the gibbons have crazy genomes. If you're interested in primates, they have all of these crazy rearrangements in their genomes that are totally different than any other primate. So we were not surprised to see that their variation was quite different than ours. But I will tell you that this likely indicates that in the great ape lineage, the VNTR is changing differently than what we see in the not great apes, as well as in like monkeys. Like, so we have sequences from owl monkeys, we have sequences from macaques, like a lot of different other monkeys that aren't apes, right? And so you can see that when I look at those sequences, they don't look nearly as stark as the orangutan and the gorilla versions, which is kind of cool.
That is really cool.
Yeah.
Thank you.
Yeah. Good question.
One more question. We have time for one more question. So once you've figured out like all the mysteries of this gene, what could people do with that?
Yeah, that's a great question. Yeah, so that is my next step. So I have been working with some computational biologists to figure out, is there a way that we can take these long-read genomes that I've annotated for all of this variation, right? And can we leverage the information in those long-read genomes to predict what short-read genomes might look like in long-read space? Basically to genotype the short reads. So we've come up with this very elaborate computational method that I did not develop, I just helped with to do that. And so I am currently trying to genotype a cystic fibrosis data set, currently trying to genotype an asthma data set, trying to get hands on an pneumonia data set. So we're trying to do these associations where we genotype cases and controls, right? Patients and non-patients, figure out what their diploid genotype is for MUC5AC and see, is there a correlation between genotype for MUC5AC and disease status? Is kind of the immediate next step. But there's tons that you can do. But you can appreciate that you can't do that unless you solve the locus first, right? There's a lot of loci out there that have been quote unquote solved, but there are a lot that still need people to sit down with the long reads to look at. So there's tons to still explore.